This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
Good evening. Why don't we try to get started. My name is Keith Yamamoto. I am a professor here in the Department of Cell and Molecular Pharmacology. I'm a molecular biologist, PhD, and also am the Executive Vice Dean of the School of Medicine and the Vice Chancellor for Research at UCSF. I'm a lifer. I've been here forever. Started in 1976. And been here ever since. I'm excited to tell you, to kind of introduce this topic that you're going to be hearing about in the next several Thursdays of precision medicine. So let me start by asking, how many of you are taking the other, one or more of the other courses? A lot of you. Great, fantastic. And then since you got your arms warmed up, how many had heard of precision medicine before you saw the topic in the course? Oh, a lot. Wow. That's interesting. Terrific. That's great. I'm happily surprised because it is basically a new concept that's being developed. It's being developed here at UCSF in a very primary way, a very kind of all-in with respect to the whole institution. And I think a lot of exciting things are happening already. And the promise of precision medicine is enormous. It's a very aspirational, sort of revolutionary change in the way that research and healthcare relate to patients and citizens. And it's exciting in that context. So that's what you'll be hearing about for the next several Thursdays. And what I would like to do tonight is kind of introduce the topic to you, say, let's think a little bit about what precision medicine is and what it isn't, why we think it's important, why this kind of revolutionary change is something that is needed. The opportunity to try to move in this direction is a new one. No one has done it. It doesn't really exist anywhere yet. But the impact of being able to do it and do it well will be enormous for all of us. It'll change all of our lives. So that's the promise. You've probably all of you, since so many of you have heard of precision medicine, probably all of you have heard of personalized medicine too. And I'll try to remember to point out what we see as the differences between the two. They're very substantial differences. And if I forget and you want to ask about that, please do that. I was counseled by my bosses up here to hold questions until the end, but I do want to encourage you, if there are burning issues, things that I'm not making clear, please interrupt at that, and at least let me know that, and I'll try to remedy that. And I guess we'll try to hold most of the Q&A type interaction to the end according to the rules here. Okay, so with that introduction, let's think a little bit about precision medicine. And what I want to do in doing that is to think of where we are now. What's the current situation? If precision medicine is a goal, what's happening now? And I guess what I would say is imprecise medicine is where we are now. And you may recognize these questions from the little blurb about the course. And I think it's an interesting way to think about where we are now and what the implications are for where we might like to be going. So what is a disease gene? If I have one, if I have a disease gene, why does it only change the risk of getting the disease? Sometimes only by a small amount. Sometimes you learn, oh, your risk doubled for getting this disease, and then you read the fine print, and the risk went from 1% to 2%. Yeah, doubled. Still pretty low. So what's a disease gene, and if I have one, why is it only changing risk instead of giving me the disease? Why is the diagnosis of disease so far from, as I say here, an exact science? Why is it sort of guesswork or statistics or something other than saying, here's your diagnosis, here's what you have? And then a question that you can kind of hold as we go through the talk, and that I'm not going to answer in the same way that I try to approach these in what follows immediately. But you can sort of think about this as we go, and we can discuss it, and hopefully the answers will kind of emerge as we go. But how could gathering information about me and then comparing it with information about many others move us toward a new level of understanding of disease and toward precise diagnosis, treatment, and cures? So imprecise medicine is where we are, this sort of statistical business of risk factors and imprecise diagnosis and so forth. Why is that? Well, it turns out that a simple and, in fact, correct and honest answer is that biology, normal biology, when things are happening correctly, it turns out to be very complicated. Okay? It's complicated for good reasons, and we can talk about those if you wish. But the fact is that it's not only complicated, but it is much more complicated than any of us who work on biological systems, the scientists, the clinicians who work in biological research, expected. Right? There are many processes and processes gone wrong, diseases, where we don't understand yet what the mechanisms are. We know that part of the complication, a good part of the complication is cartooned in this network diagram, is that these biological processes are controlled by intersecting multi-step signaling pathways. So pathways that aren't linear, they run into each other, cross-influence each other. That's what you're seeing in the cartoon. And you can see immediately where that could get us into a lot of trouble with respect to complexity. And as a function of this intersecting signaling pathways, the processes are highly context dependent. It matters a lot, whether the organism, you, are old or young, hungry or not, tired or not, right, stressed, trying to stay awake in a lecture, all the difficult things. So context makes a big difference on the way that these pathways work, right? And therefore it makes biology complicated. Now you immediately recognize from the way that I said that you want biology to be complicated in that way. You want your body to be able to respond to whether you're hungry or thirsty or not, and so forth. But it means that when you change conditions, a lot of stuff changes, right? And it makes it difficult to understand what's going on, where you are in this plate of spaghetti. Well, and since disease is merely the deviation from the normal situation, if the normal situation is complicated, then it stands to reason that disease is complicated, okay? So one kind of set of answers to these questions is simply the complexity of biological pathways. That complexity is what we want. It's obligate if we're going to be able to move around and survive in the outside world. But it makes understanding normal processes and understanding disease difficult. So there's two sort of issues that we face in sort of working our way through that complexity. One of them is this, and that is that the biomedical research endeavor is at what I call in cartoon here an inflection point. So many of you know that an inflection point is simply it's a mathematically derived expression that describes the point at which a curve changes its slope, right? And so this is an inflection point curve. And what I mean by saying that biomedical research is at an inflection point is that over the last, let's say 20, 30 years, scientists, biomedical researchers, physician scientists have enjoyed a spectacular run-up of data collection and a more and more detailed description of biological systems that has led to that appreciation that I mentioned of what the complexity is of these systems, unexpected degree of complexity of these systems that's been appreciated by collecting a lot of data and getting a more and more detailed description, whether it's looking through a microscope or sequencing the human genome, what have you. And what I'm arguing is that that what we've gained from this enjoyable upslope here that we've gained is sort of running out, and we've sort of leveled off. We've gotten to a hard place. And as we look through the hard place or beyond it, out on the horizon, we can see another enjoyable period of great run-up, right? And what do I mean by that? What I mean by that is I think that we can see that we have the opportunity ahead to be able to achieve, as I say here, a quantitative understanding of biology and disease, right, that can actually lead to an application of that knowledge in the case of what we're discussing tonight and the next several Thursdays, an application to health and health care. So that's the promise. It's fantastic. And many of you understand that, in fact, there's actually a substantial distance between data, gathering data, and understanding what the data mean. And we're all, in our daily lives in this society, swamped by, flooded by, kind of overwhelmed by data. Right? Don't know where to put it all, don't know how to catalog it, and don't understand it. Well, it's true in spades in biomedical research. Our capacity, the capacity of a first-year graduate student in my lab working for 10 weeks in a research rotation as he or she goes through the curriculum and tries to decide what lab he or she's going to work in for her PhD thesis. In the 10-week period of a rotation project in my lab, can do an experiment, a single experiment, that if we track down all the data that the student would gather, would fill entire careers, multiple entire careers. Right? So that's what the distance is between data and understanding what the data mean. So it's not trivial to get through the inflection point. But the promise that comes out the other side, the opportunities, and the ways that it can affect all of us by changing health and health care make it worth thinking about how we're going to get there. And that's really what precision medicine is about. So how do we move through the inflection point? What's it going to take? And I would argue it's two things. One is a level of integration in the sciences that we haven't seen before. To aggregate and analyze biological information, now incorporating the tools, the other sciences that can make biology this quantitative endeavor that it's going to have to be able to achieve understanding from data. Incorporating the concepts and technologies of physical sciences, chemistry and physics, engineering, and computer science. I actually should have introduced Terry O'Brien, who's sitting here in the front row. Terry is an associate dean of the School of Medicine, so works in my office there, and an assistant vice chancellor in the chancellor's office, so we work together there. So we do everything there. She's got at least as much expertise about everything I'm going to talk about as I do, and hopefully she'll jump in and fix things when I say things wrong. Anyway, the reason I brought her up this moment is I just realized in going through this list that she and I were in Berkeley a couple weeks ago talking in the electrical engineering and computer science department about precision medicine, right? Of the necessity of making this stuff quantitative. And there was a very senior, very famous computer scientist there who was really irritated that I put computer science last on the list. So, you know, I know none of you is that sensitive, but I'm just saying, it wasn't meant to say that it's the least important. I think you'll see, in fact, that it's incredibly important. All right, that's one thing. One thing to move through this inflection point is this new dimension, new level of integration that's going to be required across the sciences to make it work. And the second, in a sense, is a level of integration within the biomedical sciences, right? A new continuum, a working continuum, again, of a sort that we haven't seen before, that is deeper and broader than what has come before. UCSF is famous, deservedly so, for the level of interaction and cooperation between its investigators. This is a fantastic, unique place to work, and fantastic and unique things happen because of that, right? Even being UCSF, what we're suggesting is that we need to take it up another notch, right? To be able to build a working continuum that extends from fundamental discovery, the scientist who is curious about how some biological process works, how a protein folds. Right? Not because it's going to apply to some disease or making a biofuel or changing an environmental sensor, but because she's interested in how a protein folds. So we want this continuum to extend from this curiosity-driven fundamental discovery to translation, which you know are the scientists who want to take new discoveries and be able to apply them, in our case and in our community, to health and healthcare, to clinicians that are working with patients every day, all day, doing their dedicated work, and outside of UCSF, to the community of patients and what I just call citizens, that is, people who aren't patients, people who are well, they're not sick, and they'd like to keep it that way, thank you very much, right? And so this continuum then goes from the very basic scientists all the way out into the community and back. It's not a one-way street. It's not like we're kind of telling you stuff that you need to know, and so just listen up and behave yourselves, right? But the things that you give back to the researchers is, I hope that you'll appreciate as we go through this talk, are absolutely as important. So it's not a one-way street. It's a continuum that in which it's really an ecosystem of all of these groups, including you, really working together. And in many ways, the patients and citizens are as much owners of this process of research as are the PhDs working away with lab coats up in their laboratories. All right, so this is challenging too. It's really challenging to get through here, and to succeed, it's going to take these new forms of integration that we haven't seen before. So that's problem one. And problem two is here. What we've called the current taxonomy of disease. And we claim here that it's a problem. It's a barrier to progress. So what do we mean? Well, you know what taxonomy is. It's just a way to classify anything. You probably remember if you took a zoology course in college, that a big part of it was memorizing the taxa and understanding what the properties of various, in that case, animals are that distinguish one form from another and allow them to be put into separate boxes, separate classes. So the current taxonomy of disease, we suggest, in fact, is the second of these problems. The inflection point is the first. taxonomy in a second. Why is that? What are the properties of the current taxonomy of disease? Well, it's descriptive, right? Diseases now are classified, separated, put into separate boxes, mostly on the basis of symptoms and organs. Okay, and so we have a cardiology department that works with heart problems, right? We have an orthopedic surgery department that works with bone and connective tissue, and so forth. Okay, problems with bone and connective tissue. So disease classification based predominantly on symptoms and organs. The National Institutes of Health, some of you may know, is separated into 27 topical boxes. Aging, ear, eye, right? Cancer, cardiovascular, arthritis, various metabolic diseases, and so forth. So it's descriptive. Second, it's inflexible. That classification, those boxes, right, serve as silo barriers that separate researchers one from the other. Oh, you work on the heart, I don't do that, right? Separate our educational paradigms, right? Now it's time to understand how the lung works. And funding, I just told you the National Institutes of Health is separated into these boxes. The National Institutes of Health is where about 90% of the research funds from the federal government that comes to UCSF comes from. UCSF is the #1 public institution in terms of NIH funding. And it draws down from the separate budgets of each of those categorical institutes. So, creates silos. And it's narrow. There's really no place to put new information, new discoveries. And so that means that there's no real rationale for building that continuum that I talked about. You know, if we don't really know where to plug new information into the current taxonomy, then why get everybody together? What's the big deal? We're not going to learn anything from it anyway, because the stuff that the basic scientist does just sits over in their little corner, and the stuff that the clinicians do sit in their corner, and that's it. So that's a problem. So what can we do about it? What would be a different way to classify disease that would get us out of that second problem? What if we classify disease by mechanism? So, in a way, all of you know that would make better sense, because all of you know already, walking in the door tonight, that there are diseases that have multiple distinct mechanisms. And I've listed here 2 diseases, classified diseases, where you know already, coming in tonight, that there are diseases that have the same name but have different mechanisms. Diabetes, you know about type 1 and type 2, right? And if you know a little bit more, you know that there are multiple mechanisms for type 1 and multiple mechanisms for type 2. It's a whole family of different diseases. Cancer, each of the cancers is likely going to radiate into a big spectrum of different diseases. And the one I've listed here, we're going to spend a bit of time thinking about together, and that's a special form of breast tumors that overexpress a gene called HER2. Okay? So, in fact, what I'm going to do now is show you a video, a movie, about HER2. In fact, it's not really about HER2 as much as it is about making a drug that would be specific for to help women who have this particular form, HER2 overexpressing form of breast cancer. Okay, and so the movie is about drug development, but as you watch it, what you will see is that it's a movie about that has several key features, several reasons I'm showing you the movie. One is that it's about a disease gene. HER2, you will see, if you don't know already, is a disease gene, right? It gives rise to a particular kind of breast cancer. Right? Second, making that drug was the first example of a pharmaceutical company, in this case a biotech company, deciding to make a drug that would only help a certain number, actually a minority of patients who had the disease, and making a decision, right? And as we go through the rest of the talk, you will see that, in fact, that's what precision medicine is all about. Not trying to make the blockbusters that affect everyone, because in fact, they don't. Right? The things that are called blockbuster drugs don't help everyone who takes them. They just don't kill anyone. And so everyone gets to buy them. Okay? It helps some people, and other people it does nothing, and unfortunately it doesn't kill anyone, so just keep buying it. This was the first case of a company deciding, no, we're going to make this drug that only helps a certain set of people. That's the second reason I'm showing you. And the third reason is that the person who made the decisions to go for it, right, was Susan Desmond-Hellmann, who is the chancellor, as you know, of UCSF. And so this will give you a chance to get to know Sue Hellman a little bit. So we're going to watch a movie together. If you brought popcorn, you're in great shape. You may want to check out this iBio magazine site sometime. This is governed from UCSF. It's by Ron Vale, a professor here. It's fantastic. There's a lot of great stuff on iBio magazine.
I'm Sue Desmond-Hellmann. I'm Chancellor at University of California, San Francisco, and I want to tell you about the drug development story for Herceptin. Let me start by telling you about my background. I'm a medical oncologist by training. After medical school, I came to University of California, San Francisco, and did my training in internal medicine and then medical oncology, and had the great fortune during the last couple of years as a faculty member in the late 80s of spending time in Kampala, Uganda, as the leader of the Adult Cancer Center caring for patients with Kaposi sarcoma. And a lot of my early thinking about product development actually came from the time I spent in Uganda caring for patients with Kaposi sarcoma, and a real quest and a passion to make life better for patients. In the mid-90s, I came to San Francisco again to join Genentech, the first biotech company, and had a lot of wonderful experiences in product development at Genentech. And one of those great experiences is the story that I want to tell you about today. So let's talk about some big picture things about great drug development. And when I say drug development, I mean the entire process of bringing an idea or a concept from the labs into the clinic and then through the approval process so that the medicine can be broadly available for patients. First and foremost, and you can't do great drug development without it, you need a very deep understanding of the basic science. What's the hypothesis you're testing? What's the question you're asking? And does that question make sense, given the basic science and the fundamentals behind what you're trying to prove? The characteristics of the drug are important. If the drug doesn't get to the target, or it doesn't stay long enough in the circulation, the half-life, then you can't improve the outcome for that patient. Targeting the right patients or personalizing the therapy is the modern way to do drug development, and in part, it's the modern way to do drug development based on experiences like the one we had with Herceptin. Setting a high bar in the clinic isn't something everyone does. The risk of setting a high bar in the clinic is that you can fail. The benefit of setting a high bar in the clinic is that if you succeed, it matters a lot. It makes a big difference. People need to rewrite the textbooks, think again about what is possible for those patients. So setting a high bar in the clinic is the most inspirational way to do great drug development. And what I actually like most about great drug development is you don't do it alone. You have to work effectively with colleagues in a collaborative way, and you particularly have to work effectively with key regulatory decision makers. In America, that's the Food and Drug Administration, but there are similar bodies all over the world, and you have to work effectively with those regulators so you can do the clinical trials and ultimately so you can have broad applicability of your new product. So let me start the Herceptin story with telling you about the scientific rationale in breast cancer. And this story really rests on some critical information about the role of oncogenes in driving cancer. And this specific oncogene, human epidermal growth factor receptor 2, or HER2, is a gene that was thought to be associated with accelerating the growth of breast cancer. And the first clinical description of that growth acceleration came from Dennis Slayman and his colleagues as published in Science in 1987, and it's shown on this slide. If patients had too much HER2, either amplification or overexpression of HER2 as measured by FISH or immunohistochemistry, that was associated with a shorter survival. So about 25% of women in this trial were HER2 positive or overexpressing, and their median survival was only 3 years. Whereas the HER2 normal, about 75% of women who had a normal amount of HER2 lived a median of 6 to 7 years. That's a dramatic difference, and that matters in the clinic. That had a big impact when I was a practicing oncologist on how I thought about the outcomes in those patients. So what that meant for a patient is she could only expect to live half as long as her fellow breast cancer patients who were HER2 normal. At the time this study was done, it was particularly troubling to see this outcome because we had no remedy for this HER2 positive status in the patients. So this description was scientifically compelling, but in terms of drug development, it also outlined for us an unmet need, a difficult situation for patients that potentially we could fix. So here's the molecule that allowed us to think about addressing this HER2 positive or HER2 overexpressing breast cancer. And this is the picture or a cartoon showing trastuzumab. Trastuzumab is the USAN name, or what some people call the generic name, for Herceptin. I'm going to use Herceptin because it's less of a mouthful. But this is the monoclonal antibody. And monoclonal antibodies at the time that Herceptin was developed were thought to be kind of a pie-in-the-sky dream for a smart bullet, a guided missile targeted to certain things like HER2 that were expressed on cancer cells. And the beauty of Herceptin was that this monoclonal antibody was humanized. Most of the antibody, greater than 90%, was human. Less than 10%, in fact, in this case, about 7%, was murine. And that meant that unlike some of the early antibodies, human beings didn't reject the antibody as foreign. So Herceptin was a humanized monoclonal antibody that specifically targeted HER2, allowing us to test the hypothesis, if we direct an antibody against HER2, can we improve outcomes in women with HER2 positive breast cancer? So this is a slide that shows you the development timeline for Herceptin. And a couple points I want to make on this slide. You notice the slide is a very long timeline. It takes a lot of time to do great drug development, and that's because each step along the way, you ask and answer specific questions. Now, the first study of Herceptin was all the way back in 1991, and that's when the investigational new drug, or IND, was filed to ask FDA, we want to do clinical trials with this new concept for patients in the clinic. By 1994, the phase 2 studies were being done based on an early read that this was safe enough to give patients. In phase 2, you ask, is this medicine going to help patients? Does it look promising? And you start to figure out the dose for which you're going to do the most important phase of testing, or phase 3, which opened up in 1995. The other things that are shown in this slide as we get into the late 90s and then into the 2000s are a series of steps with the regulators, with the US regulators in terms of filing a BLA or a biologics license application, and with the ex-US regulators. And what this shows is that something had to be done that's special with a targeted therapy, and that special thing is filing both Herceptin or trastuzumab approval and Hercept test. This makes drug development even more challenging. You have to ask for approval of your product, Herceptin, and simultaneously ask for approval for your diagnostic, Hercept test. These were both concurrently approved in 1998 in the metastatic setting, or breast cancer that has spread, and then not until 2005 did FDA approve Herceptin in the adjuvant setting, or following primary surgery, as adjunctive to standard chemotherapy. Now, the HER2 testing part of the Herceptin experience was a critical part of the lessons I learned about product development when I worked on Herceptin. And what this slide shows you is a great example of why a diagnostic matters so much. It actually makes drug development cheaper and faster. And why is that? Well, this is a statistical representation of what might have been with the Herceptin study. In patients whose breast cancer has spread, metastatic breast cancer, or MBC, we knew before Herceptin median survival was 22 months. So 50% of patients had died by 22 months when you studied first-line metastatic breast cancer. Let's say that we expected the benefit of Herceptin to be to extend that survival from 22 months to 27 months, or a 5-month improvement in survival. Well, if everyone in the study, or 100% of the patients, were HER2 positive, that 5-month difference would take about 1,250 patients and 52 months duration of study. If we didn't have the diagnostic test, and truth was that only 25% of our study population had HER2 overexpression, that study would have shown only a 1.25 month difference and taken 349 months and 11,000 patients. We couldn't have done that study. That study wasn't possible. So the diagnostic allowed for a smaller patient group and a faster study. And here's the answer to the question, does Herceptin help women with metastatic breast cancer? The great news is the answer is yes. In fact, as we had predicted, the median survival extended from 20.3 months to 25.4 months, a 25% improvement in survival in women with metastatic breast cancer, all of whom were HER2 positive when treated with Herceptin. Even better news came when we studied Herceptin early on, the adjuvant setting. The time to first distant recurrence doubled. The time that women stayed in remission doubled when Herceptin was given just after surgery. And this news was so dramatic that it got a standing ovation when it was presented for the first time. In my experience, the first time I've ever seen a standing O at a medical meeting. Great news for patients treated with Herceptin. Here's the New England Journal article that talked about that great news for women in the adjuvant setting, HER2 positive, treated with Herceptin. And the most important thing that I want to point out in this New England Journal article is the last line. This will completely alter our approach to the treatment of breast cancer. Now you have to ask, is the patient HER2 positive? Can you help women by targeting this scientifically based new therapy at what could become the Achilles heel of the cancer? And the answer was yes. So let me come back to the key things. We understood the science, we had the right drug, we targeted the right patients, set a high bar, survival in the clinic, and we worked all the way along the line with the Food and Drug Administration. And the best thing about working on Herceptin and the experience I had was I know now hundreds of thousands of patients are going to benefit from this work. And that's the best news of all in great drug development.
All right. So I think you saw a couple of things there. I hope you did, besides the fact that Sue's awesome. I mean, making that decision at Genentech was not just, as I said, making the decision to make the first targeted drug to a minority population of patients, but using this monoclonal antibody strategy, which many people said was just destined for failure. So it took a lot of courage to go ahead and do that. So that's what she's about. Okay, so what did you see? Well, you saw that HER2 is a disease gene, right? That HER2 overexpression doesn't predict breast cancer, right? It's not that everyone who overexpresses HER2 has breast cancer, but rather predicts an increased risk, right? So it's not a done deal. Moreover, breast cancer can occur without HER2 overexpression. There's a big population, you saw, 75% of women with breast cancer who don't overexpress HER2. So it's neither necessary nor sufficient, right? But it's still very definitely a disease gene. But for the women who have breast cancer and HER2 overexpression, Herceptin, the medicine that was made that Sue described, increases the time of survival. So you can make a targeted therapy. But there's a second but, right, that you also saw in the data that she showed. And that is that Herceptin doesn't cure breast cancer. It extends the life expectancy. Why is that? Because biology is complicated, because disease is complex. HER2 overexpression provides a mechanistic classification, a new kind of taxonomy, based on mechanism of breast cancer. But because disease is complex, Herceptin alone is not a cure, even for that specific class of patients, of women, who overexpress HER2. It's more complicated than that. There's more stuff going on. And a goal of precision medicine is to sort that out so we can achieve precision in being able to say, here's what you have and here's what you need. So it's a huge step that what Sue did represents, but you could see from what she presented, right, that it's not, we're not there yet. So that's certainly one great reason for classifying disease by mechanism instead of symptoms and organs. And here's another one. And this one you may be less familiar with than what you already knew coming in, and that is one disease can have more than one mechanism. And the second reason is that one mechanism can be implicated in more than one disease. My favorite example of this is a set of diseases in what are called primary cilia, which is what you're looking at here. So you're looking at electron micrographs of primary cilia. Primary cilia are just little hairs or projections that extend from every one of your cells. All of your cells have a primary cilium, usually one, that sticks out. So why is this interesting? It's interesting and important because there's a set of diseases that come from defects in making the cilia, in synthesizing it at all, or in assembling it in the way that it makes the little hair thing correctly, and so forth. Right. Now look at the diseases. Situs inversus, some of you may have heard of before. It's a fascinating disease in which your organs are on the wrong side of your body. Polycystic kidney disease, obviously, it's a very serious defect in your kidneys and kidney function. Hydrocephalus, water on the brain. Retinal degeneration, you can figure that one out. Polydactyly, wrong number of digits, extra digits on your fingers. Wait a minute, those diseases don't sound very related. They're not, except they are. They're all caused by defects in making the primary cilia. So there are people, scientists, doctors, working on these separate diseases that don't talk to each other, but they're all working on the same thing. They could all learn from each other. So that's a second good reason to stop classifying disease by organs and symptoms and start classifying by mechanism. So the idea is that a mechanistic classification of disease could capture and use more information for research, education, and healthcare. Great. So now we've really gotten somewhere, right? We start out with these two problems, figure we've got to work our way through the inflection point, integrate in new ways, and change the way we classify disease. Excellent. But how do you do it? You don't just say, let's stop having a department of orthopedic surgery and make a department of cilia. That's not going to work. It'll just be another silo. So it's great to say it, but how do you do it? So the goal is here, is to begin to pull together this vast data flood that we're all facing, integrate the rapidly expanding, maybe somewhat randomly too, rapidly expanding range and detail of biological, environmental, behavioral, experiential stuff, information, with three goals in mind. Make that basic scientist able to ask and answer questions that she didn't think of before because of this availability of all this now integrated and organized information. Create a new system for disease classification that's mechanism-based, that pulls together the right people and has them communicating with each other, and enable better medicine and reach toward precision medicine. Fantastic goals. So how do we get there? So here's the proposal. Here's kind of the idea of precision medicine. That is to talk to our friends, the computer scientists, and build a vast, what we call information commons, which is just a single place where all information about biological systems resides, or actually can be accessed, to be frank. And then to mature that massive information into what we call a knowledge network that I'll define for you. that can correlate and align disparate information and extend the acquisition and use of data from the lab to the point of care to the course of daily life. This is where you come in. That's the proposal. Now, where this started was in an interesting place, in a study that was done by the National Academy of Sciences that resulted in a report that published at the end of 2011 called Toward Precision Medicine, Building a Knowledge Network for Biomedical Research and a New Taxonomy of Disease. And there's a URL there. It's up on the National Academy website. Now, many of you may have heard of the National Academy of Sciences, but I'll bet a lot of you don't really know what it's about, what it's for, what its function is. It's a nice honorific society. But in fact, it was founded by none other than Abraham Lincoln during the Civil War. You know, kind of in his spare time while the country was, it's kind of a classic failure to focus, I think. He decided, oh, this is a good idea. let's make the National Academy of Sciences, because we need a private, independent, unbiased source of scientific advice to the government. So the National Academy of Sciences is a group that is charged by the government with providing advice about scientific matters. Okay, and that's gone on since 1865. I happen to be chairing one of the boards called the Board on Life Sciences for the National Academy of Sciences, and we received a request from a government agency that said, tell us about whether it would be a good idea to make a new taxonomy of disease. And so I took this idea, took the letter to the board meeting, and we discussed it. And frankly, when I received it, I thought, this is not interesting. We're already studying the mechanisms of disease. This is what we do this in my lab and in many laboratories here at UCSF. There's a lot of mechanistic stuff going on. I'm not worried about this. But the more I thought about it as the meeting approached, the more excited I got. So by the time the board was in front of me, I was quite jazzed about the whole thing, and we decided to move ahead. And it's what became precision medicine as the committee began to define it. Now, when you're a board chairman at the National Academy, you don't do the reports. You decide whether to move forward with the report. You sort of craft it into a shape that you think makes some sense, and then you go find a bunch of really, really smart people to do the study and write the report. So a key part of that job is, once the trigger is pulled, is finding the right people, especially the right people to chair the committee, to really give it direction, and so it can have the chance to have a meaningful impact. And so I was excited enough about this that I was determined to get, as chairman, co-chairman of this committee, Sue Desmond-Hellmann. Because I knew, first of all, she would do a fantastic job, which she did. And secondly, that if she co-chaired it, she would get excited enough about this that she would want to make UCSF kind of prime ground for being able to start this endeavor and see what we could make it go, to have a big impact here at UCSF, but then in doing that, hopefully have a big impact nationwide or worldwide. And so that's what's happened. Terry was Sue's staff person in the course of preparing the report. I was on the committee and was involved in it as well. Okay, so that's sort of interesting in its own right. So next time you hear about something coming out of the National Academy, you'll think immediately of Abe Lincoln. Okay, so now I'll sort of just say in these three phrases what precision medicine is. We sort of spent the time so far sort of in a way saying what it is not and what we aspire to because of what it's not. So let's get specific about what it is or what it will do as sort of the promises as we see it. Precision medicine will aggregate, integrate, and analyze biological information drawing on vast collections of data to produce accurate health advice and predictions, disease diagnosis, tailored to the individual. So it's using massive amounts of data, right, to be precise about diagnosis and treatment of you, specifically you. Precision medicine will be enabled by this giant computational thing, fantasy, computational information commons, within which correlations produce a knowledge network. So links between bits of information in the information commons form bridges that then produce a network of knowledge, generating new hypotheses for the lab scientists, illuminating mechanisms of disease for the clinicians, establishing links between diseases thought to be unrelated because of the old way that diseases were classified. And precision medicine will revolutionize development and deployment of therapies, as well as principles of clinical practice, by allowing stakeholders to both benefit from and participate in the research, health, and healthcare enterprise. And that extends, of course, to you. Okay, so kind of big aspirational stuff. How does this knowledge network work? So you're all familiar with GIS, Geographical Information Systems. Even if you don't know the name, you know from Google Maps what it's all about. Google Maps is GIS. It is layers of information of different sorts that you can see here, whether it's postal codes or transportation maps, that are stacked on top of each other. And because Google Maps, because in Google Maps, the information is all aligned by geography, you can drop a plumb line through any point on these surfaces and bring together information on transportation, land use, census tract structures, and so forth for exactly that position, just by dropping a straight pin through. The information commons for health and healthcare and research that we're talking about is similar in one way and different in one way. The similarity is that it's, again, stacks different kinds of information up in layers, really disparate kinds of information, starting with information about individual patients, right? Other types of patient data, and then you see some ohms, the genome, the microbiome, some of you have been hearing about. These are the microbes that live on the surfaces of our body, whether those surfaces are on the inside of us or on the outside of us, that are incredibly important. There's more you have more microbe DNA on you than you have your own DNA in you. The human genome sequence and so forth. And then a bunch of stuff that doesn't fit into these kind of nice, objective, lab experimentally determined stuff, like signs and symptoms. The exposome, that's sort of a new one. Stuff in your environment that you get exposed to, especially as mentioned, as calculated over the course of time. Things may change. You grow up in the middle of Detroit and live in Northern California and so forth. Okay. In this case, here's the difference. And that is, there's no common orientation point for this information. You can't learn anything by just dropping a straight pin through. In fact, instead, you've got to do the hard work. You have to do research to be able to understand relationships between any data point on any of these layers and a data point on any of the other layers. It's not just a straight line. There's no common orientation point. And discerning those relationships is what building a knowledge network is about. So I'm going to come back to the details of what the knowledge network is like and how it might be built, right? But let me show you first the way that the committee imagined that this thing could have a big impact. And that's what's cartooned here. So here's the information, in the middle is this big block of information commons and knowledge network. And really all this cartoon says is much less complicated than it looks at first glance. All of this cartoon says is a bunch of data shows up from any of these sources. So clinical research in the hospital, basic research in my laboratory down in Mission Bay, characterizations of specific molecules like cilia, the electronic health records, your electronic health records, and so forth. All of that gets dumped into the information commons. right? And then in layers. And correlations are made that then comprise the knowledge network. And from that, new knowledge comes out the bottom. And for the basic scientists, can form new understanding of mechanisms, can form new hypotheses that lead to new experiments, and more data gets generated that goes into information commons. For the clinician, right, new ideas about diagnosis and therapy, new ways of thinking about the outcomes of disease and treatment, right, that can turn into more clinical studies and clinical trials that then generates more information that goes back into the information commons, and you get this swirl that's all being fed through the information in the middle, right? And so now all of the information is being used, is what we're looking for, right? And it's being used because there's this new integration that brings all the different kinds of science together. We're using these big high-end computational tools and other quantitative studies, and using that new capacity to generate this new taxonomy of disease and new generation of knowledge. Right? So that's the idea. That's the hope. Okay. So now let me go back to this notion of building and even using the knowledge network, which extends to you using the knowledge network. So this is not a knowledge network. This is a disease gene network. But I think it gives you an idea of what's in those flat layers, right? And so in this case, what you're looking at, each of the dots represents a gene. And if you're close enough to see the code, the different colors represent different disease areas, and so the genes that have been implicated in causing or being correlated with the incidence of a given disease are clustered together. And you can see some of them cross over and bridge over to different disease types. So there are genes that are involved in more than one disease type. We're familiar with that now. We understand that, right? And so this could be, and then there's a bunch of genes that haven't been assigned yet, that haven't been correlated yet with a particular little node. So this could be one of the layers that you then turn on its side as the genome, in this case the disease genome, and forms one of these layers, and then you just stack other stuff on top. So the proteome so the genome, of course, is the DNA, your DNA sequence. The proteome are the proteins that are made maybe in a particular cell type in your body, maybe all of the proteins that you're able to make. Metabolome, this is an interesting one. These are all the small molecules that are out swimming around inside of cells or inside of your bloodstream, any particular place that we choose to look. Small molecules are interesting because they incorporate information, the processing of molecules, not only by your genes, but by the genes of your microbiome, by other things that you run into that can process chemicals and so forth. So it's sort of a complex integral, in a way, of a lot of information being brought together. Very, quite interesting. Here's the microbiome, and then effects of the environment, population studies that are derived from population behaviors that are derived from epidemiology, and your patient medical record, and we can make a whole series of stacks that go out the top of the building, right? So there they are, lined up. There's correlations within a layer, and what building a knowledge network is about is drawing now correlations between layers. So interlayer connections are established by teams of scientists or clinicians that use these different tools that give rise to the genome sequence of the proteome and so forth, working together and talking together and doing things that allow them to begin to make correlations. So the links between the genome, your DNA, and the proteins that you make, there's a lot of pretty sturdy relationships that we can already draw. We know what protein a given gene encodes, for example. But as you go up the ladder, the correlations become kind of softer and fuzzier and less certain, right? But they get drawn anyway. That's the idea of a knowledge network. And it gets matured and better as the research goes along. And so you start with what you have, right? And you go from there. So for example, my laboratory is full of molecular biologists. And so we're really happy working down in this great experimental objective stuff. If a population or a behavioral scientist were to walk into my lab and say, "What are you guys doing? You know, don't you know that people's behavior or their environment or what they're exposed to is really critical for disease?" People in my lab would look up and say, yeah, we know that. It's really important. We got that. You know, please go away. We're trying to do science here. And that's a silly attitude. That's the kind of separation that we're talking about, that we got to stop, right? Right? Because we all do know that the environment and exposure and experience and emotion affect whether we get disease or not. And it's time to figure out what those links are. So that's what's going on here. Here's an important point. The networks grow and change as connections are added or subtracted. So imagine this one. Here's a protein in your proteome, this guy, where there's been kind of a loose correlation made between some element in the environment, right? And so we get to draw that line, right? Further research goes on, and oh, look, now we understand that there is something in the microbiome that also tracks to that production of that protein. So we get to draw a new line, right? And maybe future research will show that that component in the environment that we've noticed was a direct result of the presence of this microbe in which this line was drawn. And so now that first line will get subtracted. Right? So when things change like that in this system, it's because we're getting better. The knowledge network is maturing, it's getting better because of more research. Additions, subtractions can both come from advancements in understanding. So the knowledge network then serves these three key purposes. Enables new discovery, sets up new hypotheses by making connections that we didn't expect before, that says, let's do an experiment and figure it out. Is that environmental component, in fact, the new microbe that we discovered? Can improve clinical care because of new knowledge coming out of the bottom of these systems that is the result of experiments that have brought together not just clinical studies that the doctors will always have always had, but now the mix of basic science as well. And it informs this taxonomy that changes the way we classify and think about and study disease. So here's an important point, and that is that as the committee thought about this, a critical component was that the knowledge network is accessible to everyone. It would be visualizable on your computer. Visualizability is key here. The brain doesn't have very many ways to access and think about, process vast amounts of knowledge. And so there have to be sort of crutches, and visualization is a key one. And we're working with big-time data visualization people who do this for a living to say, how are we going to do this? How are we going to take vast clouds of information and make it visualizable in a way that the brain can accommodate it, and then dive in as deeply as you want to go to then understand the key publications in this area, what's new going on, who are the people doing the work, right? How are those basic bits of information linked to disease and so forth, for, a long string of stuff for the ways that accessible information can be processed and used by anyone. So it means that you can sit down then at your computer. The cloud information will look nothing like this. This is just what Terry and I were able to draw with our rudimentary capacities for PowerPoint, much less computation. But it means that clicking on a node or connection or sweeping out a zone with your cursor, would bring together bits of information, a collective of information, right? And in fact, if you're a scientist, define a team of people that you might want to work with, right? By sweeping out the zone and saying, who's doing this stuff? I can't do most of the stuff I can't do. I could do a little bit of molecular biology. But now that I've swept out this team, this zone, there's a team of people that if I work together, we could do things that none of us could do by ourselves. Or if you were sitting at home, sweeping out a zone, you would say, who are the people, let's say at UCSF, that are working in this area, coming at the problem from many different directions? So the network connections then suggest hypotheses and mechanisms, and a key to this is data. It depends on a lot of information coming in. That the more information there is, the better and the more precise the conclusions. So the nodes and edges depend upon data contribution from patients and citizens. You have to understand that and be motivated to be able to contribute information knowing that it could affect your health, not just the health of your grandchildren, your children, but your health. That new connections would carry work into new areas. This happens all the time in science now. Certainly in molecular biology now, we isolate some gene that is responsive to some signal that we're giving, and it's a gene that we don't know anything about, but other people do. Right? And so the knowledge network then says, here are the people that know a lot about this. Go talk to them. So it carries the work into new areas, and the network reveals potential research teams, new patient groups, and disease subclassifications. It would be enabling for all stakeholders, patients and citizens, academic and industry research, bringing together the fundamental stuff that goes on at UCSF with some potential products or medicines that can be made at Genentech in the way that we heard from Sue. Changes the way we think about education. Shouldn't we be bringing our students together that are now taught separately in medical school and in graduate school, looking for an MD and a PhD? Because in fact, if they work together, they can go after problems that we can't approach right now. And they can find each other through the knowledge network. And it checks our funding and regulatory agencies. You heard from Sue about the FDA, right? And thinking about this, right now, by the time a drug gets to phase 3 clinical trials, a drug company has spent several hundred million, sometimes close to $1 billion. And in phase 3, if a couple of patients drop dead unexpectedly, the FDA flunks the drug and you're back to nothing. So where do all those costs go? They go into some drug that does succeed, and they make drug costs really, really high. What if, instead, we either knew enough, like Sue did, to be able to construct a trial that would be filled with patients who had 100% HER2 overexpression, so they would respond and they could get away with 1,250 patients in a 4.5-year trial, instead of not knowing and having only 25% of the patients HER2 overexpressors and 11,000 patients and 29 years. Don't think a drug company's going to do that. That's what she said. Of course, she's right. So enable all stakeholders, patients, funding and regulatory agencies, maybe we would change the way that NIH is structured. All right. So, so now let me tell you what it is that we're doing here. If you hit that website, the National Academy website, and look through the report, or look through the, how long is the executive summary? We have copies. Awesome. Okay. There are copies. It's just, anyway, it's just a few pages. The executive summary is only a few pages. You will see that, in fact, the conclusions of the study is almost embarrassingly simple. There's three big conclusions. There's a bunch of conclusions that are written out, but they really fall into three big categories. One, good idea to reclassify disease by mechanism. Two, to do that in a way that will have an effect, an impact, build an information commons and knowledge network. Really hard, but that was the second conclusion. Third, this is going to be really complicated. So there should be pilot studies done around the country, around the world, right? For institutions or individuals who think they can do something that would make a contribution to assessing assessing the best way to move forward, right? So we can't start this at full scale, country or internationally, and so there should be pilot studies done. So we're doing one. We're doing one that, as far as we know, is unique. We're the only ones doing this kind of pilot study. Right? There's a bunch of precision medicine institutes that have popped up around the country since the report published, right? And they are all doing a subset of this second element that UCSF is rolling out. And they're all studying genomic medicine, linking your genome information to your patient medical record. Very important, very difficult. Don't let me demean that goal. But the efforts in all the other places we're aware of are limited to that. UCSF is doing this whole spectrum. We're trying to build UCSF-sized model of the whole national experiment to see what we can do. Knowing that, right, being the first ones out, we'll probably step in some holes, right, that have to get fixed. But maybe we'll be able to set best practices for a national level model. And as we do it and make it work here, it will have an enormous impact here on investigators, clinicians, and patients and citizens. So we have 6 elements of a so-called precision medicine platform. Those 6 elements include stuff that you've been hearing about: knowledge networks, right? Basic discovery, clinical discovery, and so forth. Those 6 elements work together, the first 5 work together to feed information into the information commons that matures into the knowledge networks, generates new knowledge, and so forth. So it ends up giving rise to this. We have this precision medicine platform with its 6 components, information commons and knowledge network being one, that generate new knowledge as the stuff gets pumped through the mill, as we looked earlier, in which the new knowledge can then be used to cycle again. A critical component of this, and sitting at the very top, is information coming, data coming from patients and citizens. And some of the new knowledge can get fed back out to them. So now I'm going to give one slide each of those 6 platform components to tell you a little bit more detail about what we're doing already or intending to do moving forward. I'm not going to give a lot of detail because of time, and because, in fact, some of these elements are going to be detailed in subsequent lectures in this course. Okay. So basic discovery we've already been talking about. That's just basic science that goes on here in my lab and many others. Conduct innovative investigations of fundamental biological processes. The work that goes on at UCSF is distinctive. This is a terrific place. Recently, meaning the last few months, a new discovery was made that took the scientific community by surprise. And that is that there are small extracellular RNAs. Okay? RNA is a nucleic acid. It's thought as a resident of the inside of cells. And the discovery was that for some, undiscovered until now, there are some RNAs that are getting spit out of cells and somehow survive. RNA is thought to be a very fragile molecule. They somehow survive by getting packaged in little vesicles, little spheres, and they move around and they can go into other cells. So it's nucleic acid, so genetic information being moved from one cell to other cells in ways that was totally unexpected. How these things are released, what they're doing, unknown. So the NIH has just granted, just funded, 5 big exploration grants. 2 of those 5 came to UCSF. Mike McManus and Robert Blaylock. So 2 out of the 5 of these big Fortran grants to figure out what is extracellular RNA, what the heck is it doing, how does it get out, where is it going, what does it stand for. It's going to be important, for sure. There's already very clear indications of that. But how it's important, what the implications are for understanding fundamental mechanisms, understanding disease, not known. Develop methods and technologies that improve rates or resolution of analysis. A new technology, CRISPR-Cas, doesn't matter what it means, what it stands for. What's amazing is what it can do. A new technology that offers a revolutionary way to edit the DNA in your cells, in your genome, right? Human genome or other animal genomes. And so scientists here at UCSF and collaborating with scientists at UC Berkeley have put forth a proposal to use that new revolutionary method to knock out in cell lines every human gene and see what the consequences are. So something that was unimaginable before this new technology came up. There was no way to do that. Moreover, you suddenly can think of a whole zoo of things you could do with this technology, right? If we wanted to make an animal model of a specific disease, right, a change of a particular amino acid in a cancer gene, now you can use this technology to do that. You can make a mouse that has exactly the same mutation that we know leads to human disease and ask, how accurate a model of that disease is it? If it's really accurate, we can use that mouse to carry us a long way in understanding the mechanism, deriving and testing therapeutics, and so forth. Establish new connections between ideas and observations thought previously unrelated, a key component of basic research. Clinical discovery. Conduct innovative clinical trials that integrate clinical and molecular information, bring together these two worlds. Build a virtual data warehouse that brings together the basic research information together with clinical data. And then all of the complicated stuff that has to get thought of and taken care of in order to be able to move, to do research on humans, to be able to move therapeutics around and test them while assuring privacy and so forth, is the charge of this group. Let me just expand on this with one pretty cool set of experiments. Keyvan Shokat is a wonderful scientist here. Actually, he's a chemist that I was involved in hiring when I was back when I was a department chairman in the 90s. And he's done an amazing thing. So what you're looking at in this table is a set of genes that cause cancers, oncogenes, right? something about the types of tumors that they cause, and so forth. And some of those cancer-causing genes are primarily causing their cancers by having particular mutations, mutations at particular locations, amino acids, across their coding sequence. When people make therapeutics, right, if you make an inhibitor to an oncogene, you figure out some way to slam, if you're really good, you think of some way to slam that specific oncogene. Right? But remember, the genes are not there to cause cancer. They're doing something important in the body. So what you really have to do is find what's called a therapeutic window. You have to do enough damage to this gene product so that the cancer stops growing, but not so much damage to it that you stop. That's the therapeutic window. So basically, cancer therapy in general is basically poisoning the patient, but not quite enough to kill the cancer, but not enough to kill the patient. Kayvan said, this is an opportunity. Look at these two oncogenes. 20% of the time, they cause cancers, really bad ones, in which they have over and over again the same mutation. So he figured out a way to make drugs that would affect only the mutant oncogene. And so now the therapeutic window is wide open. The normal gene doing its normal things is unaffected, and the drug goes after only the cancer-causing mutation. Incredible wizardry of this guy. One of them is in clinical trials, and one of them he just reported in the literature. Incredibly exciting. If you look at those cancer types, you can see it'll be pretty good to be able to go after those. Talk about bringing together clinical and molecular information. This is what it's about. Omic medicine. Collect and analyze human subject genomic and other omic data in validated, secure environment. You have to be able to move this information, very much front-end, next-generation laboratory stuff, into the patient environment where the information will be used. Some really fascinating stuff. Robert Nussbaum, looking at the value of gene sequencing in newborns. This is this capacity is there. How much will it help? How much will it be better than blood tests? What do we do with the information that we get if we get new, much deeper, detailed disease information for which we have no way to treat, and so forth? New study that is undergoing. Another study. Elizabeth Blackburn, Nobel laureate here that you've probably heard of, or maybe many of you know or have heard her talk, basic scientist. Peter Carroll, as a surgeon, chairman of the urology department, bringing together people who know and think about very, very different things in a single study that looks at the effects of various lifestyle things, diet, exercise, stress management, social support, on telomere length. That's what Elizabeth Blackburn got a Nobel Prize with. These are the little caps at the end of the chromosomes, where it's been shown that as you age and get older and get diseases, the chromosome little caps get shorter and shorter and shorter. And what Liz and Peter Carroll have found is that in a cohort of prostate cancer patients that Peter Carroll has, right, that positive lifestyles actually lengthen the telomeres. And it's thought, and there are still early days on these studies, that maybe it will result in less exposure, less risk for chronic disease, maybe even longer lifespan. Who knows? We'll see. Coordinate the sequencing efforts to be able to move them into government-certified patient environments, assure these privacy compliances, the things that we were talking about, and build out a set of education programs. And Robert Nussbaum, who's chairing the Omic Medicine part of the platform, has already developed some really interesting courses, including some massive online courses, these so-called MOOC courses available to anyone. Institute for Computational Health Sciences. It's already been inferred what a gigantic computational challenge building the Information Commons and Knowledge Network is going to be, and we're in the business of, so we acknowledge that we don't have a computer science department here, but we have friends. That's what Terry and I were doing at Berkeley, talking to the computer science department. And we're going to build out our own that's specially tailored to be able to build Information Commons and Knowledge Network, especially tailored to trying to make precision medicine work. Center for Digital Health Innovation. Again, this is now the link, in many ways, one of the closest links to patients and citizens. Digital health is a revolution that's coming at us. There are tools being built by companies large and small, not even companies, just app builders in their basements. And it's the job of this center, and Michael Blum, who's the director, to motivate, partner, develop, implement, validate tools that collect, aggregate, analyze, and report health information in the course of normal life. And increasingly, we're going to not only have apps on our smartphones, but little patches that we're wearing and so forth that will report health information. Any bad trends will go to some big central computer somewhere, and maybe your primary care physician, and certainly to you, and so forth. So these things are coming, and the opportunities, as well as the ethical issues and privacy issues, of course, are in front of us. Aggregate expertise of the people who think about building such tools. We have an Office of Innovation, Technology, and Alliances here that includes an Entrepreneur Center that says for the UCSF faculty that are excited about actually building devices that will be able to test things that are that they know are important from their research. We want to give them the capacity to try to move the work into the private sector. And organized clinical trials. There's a set of those ongoing. You can hit the website of CDHI if you want to take a look at them, but in fact, later in this course, you're going to get a real treat and hear about some of these in detail. There'll be a whole session that's dedicated to digital health. And then finally, the Knowledge Network. We have made the decision to build the knowledge networks out from neurological disease and cancer. We have good reasons for doing that. Notably, fantastic work here, buttressed with Nobel Prizes in both areas, but also fantastic, amazing stuff done by young scientists. But if a network is a network, it'll spread and cover the universe. It's just an organizational way to start, and I think we start with two very, very important areas. Visualization, as we said, is critical, and combining the expertise across that continuum of knowledge and research is going to be key. And of course, this extends out to take advantage of the fact that we're sitting at the top of Silicon Valley, and there's some pretty smart people down there that seem to know something about computation, and we're talking with them as well. So that's the pilot study. We think that it puts together a platform that best utilizes and makes powerful use, in fact, of these three critical parts of the triangle of basic research, clinical research, and patients and citizens' information and data. So that really brings me to the end then, and I'll come back to this in a slightly modified form, that research in healthcare are at an inflection point. That it's been great. We've had a great run of being able to collect data and describe and realize the remarkable complexity of biological systems. And we've got an opportunity to work our way through this inflection point and reach a quantitative understanding of biology and disease that will take us to a point of being predictive, preventive, right, and precise about care. So reaching toward precision medicine then, integrate across the sciences in ways that will really advance both fundamental discovery and determine diagnosis and treatment of individuals using lots of data, and developing this working continuum from fundamental discovery to translation to clinical research, to clinical practice, to patients and citizens, and back. Okay, so with that as a backdrop, you're in for a real treat in the next few Thursdays. Next Thursday, you'll hear about this research continuum, one that is definitely co-owned by patients and citizens. Martin Kampman, a brilliant researcher in Jonathan Weissman's laboratory, will talk about how there's some basic research experiments that are being done that really have a direct bearing on multiple myeloma. Melissa Paoloni, Director of Advanced Clinical Trials and associated with Quantum Leap Healthcare Collaborative in San Francisco, will talk about a very innovative set of clinical trials that are going on in breast cancer. The next Thursday, the Knowledge Network. How do we, let's drill down on this sucker and talk about how we actually build it in a way that will merge and integrate life science and disease information. Pierre-Antoine is a researcher that works on multiple sclerosis and has built a fantastic data network, so-called switchboard, that brings together information from basic to applied to looking at longitudinal brain imaging studies for MS and bringing patients and citizens into that mix. Catherine Rankin is an associate professor in our Memory and Aging Center, who will talk about, again, bringing together imaging and omic and other sorts of front-end research information to be brought to bear on issues of memory and aging. And then November 7th, you'll hear about privacy, security, regulation, and voluntary data sharing. What's that going to look like? It's sort of a new world if you think about how you might want to try to make this work in a way that motivates you to provide data where you feel secure enough that you'll actually do it. And Claire Brindis and Dan Dohan and Minnie Kalon, coming from different directions, will consider some of those issues and opportunities with you. And then the next Thursday, November 14th, you're going to get a real treat from Rainier Sawyer, who will talk to you about digital health and this incredible revolution and opportunities and dazzling tools that are being developed that can do all sorts of things that have the potential to be able to really get a head jump on disease, that will be able to begin to monitor things in you and on you in a non-invasive part of your normal life, where not only you won't have to wait to go to the hospital, you won't even have to wait to get sick, because trend lines will change and you'll be alerted ahead of time. And then finally, Hein Sepulveda, who's our Executive Director of Global Health Sciences, who's here actually right tonight, we'll talk about the fact that, in fact, the community is not, for precision medicine, is not UCSF or California or the United States, but the world is the community, and what the implications are in global health for precision medicine. And that concludes the course and concludes my presentation tonight, and I really thank you for your attention. Happy to take questions. Questions?
One place where this amazing picture is being played out in reality is Fukushima Prefecture. When the tsunami came and washed over the reactor, it had the effect of washing away the silos, very strong silos in that society. And suddenly, highly competent, highly educated professionals are almost accidentally shoulder to shoulder with people they never would have spoken to before. And they're astonished by how far they're coming in a short time. I don't think they've had time to publish, but they're facing a deep crisis. I don't want to minimize that. But they're also having this exciting experience of exactly what you're presenting.
Exactly right. And so this person's making the point that in Fukushima, after the disaster there, the silo walls were forced down. And people were forced to get together and talk and work in common on problems, and that's exactly right. I visited Tulane, where after the disaster in New Orleans, exactly the same thing happened. And I heard the same kinds of stories about the communities being pulled together and this, remarkable discovery of each other, and the fact that there's a lot to be said to each other, and it's exactly right. So basically, this is really an attempt to be able to do that without the accompanying disaster. Question?
Well, capitalism can either erect silos or break them down or both. Do you envision there's going to be any problems with, we had people trying to patent genes and stuff like that?
Yeah, I mean, I hope embedded not below the surface here are the considerable questions about security and privacy and ethics that are really driven to the surface by bringing together a lot of information and using it in common. Just to make it really explicit, because your point is exactly right. I said, isn't this great? All the information will be accessible to everyone, right? Well, what about insurance companies? What's going to make them behave? If insurance companies have information about you, right, that indicate that they don't want to insure you, what's going to make them behave? And I'll tell you, I think there's only one thing that will make them good stewards of information. And that is if they can become convinced that if they're good stewards, their costs will go down. Right? I believe that that's true. We're working with some health economists to begin working on modeling this. And we can go, I could take you through that, but I believe it to be true that, for example, look, what if we could answer the following question? What if we could reduce by half, that's not very ambitious, reduce by half the number of prescriptions that are given that don't help you, and reduce by half the number of clinical tests that are given to you that don't help you, that you don't need, right? What would that do to the health economy in this country? Right? So there's some health economists that are working on that kind of question.
Damage a lot of pharmaceuticals.
Damage pharmaceutical, maybe medical schools, right?
Yeah.
How about that?
So you might be helping insurance companies, but you're going to bore someone's eyes.
So yeah, someone's going to get hurt. Okay, fair enough. So those are the kinds of things that I think we need to be considering. The issues, it's a good point, and the issues are incredibly complex. Questions? Yes, I'm sorry.
So I work with people helping them navigate through complicated medical situations. And what's really exciting to me about what you're talking about is the biggest complaint that consumers have about medicine is that nobody talks to each other, that the clinicians, the specialists are. So I'm looking at that model going, what if we totally changed it? What if we, instead of having rheumatology over here and orthopedics over here, what if we actually had clinics where they were all together so that when somebody came, they saw people all in the same pod? What would that do for communication and problem solving?
Yeah, and you're going to hear about some experimental clinical trials that essentially do that. That kind of make one-stop shopping, and that's incredibly exciting. As a scientist, I have a fantasy that research institutions like UCSF, in some future time, if this were to work out, would look entirely different, where researchers, whether we're old faculty members like me or new trainees in my lab, won't be sitting in a lab working for years and years and years on a single problem, but they'll all be members of, let's say, a half a dozen teams that have come together because of this knowledge network, that are working on things that are incredibly exciting that they never could have imagined being able to work on before. And those teams are constantly changing. They're finished with one project, they're happily jumping onto something else they also didn't know anything about before, right? It could really make it could make the research and the organizational structures that are doing the research as dynamic as the opportunities are in front of us. All right, I'm being told we better stop, so I'll take one more question.
Yes. I have a friend who sent a sample of saliva to an institute, I don't know the name, and she received a report back about her genotype, risk of disease, and so forth. Is that a good way to collect information?
There are a number of crowdsourced experimental programs and companies like that right now that are pulling together information. There's a lot of information in saliva, right? There are three basically human materials that are kind of non-invasively available. Saliva is one of them. You can figure out the others. And there are tremendous opportunities there to learn information about yourself that we're presently not collecting. Now you could say, what's the value of just kind of, let's say the metabolome, cataloging all the small molecules in urine, right, for a whole bunch of people? Well, if you couple that collection with gathering other sorts of information about their health, when they get disease and how long it lasts, when it's first diagnosed and so forth. And then you go back and look at those small molecules and say, were there any changes? When was that? Oh, look, it was 5 years before it was diagnosed. Right, and so now you know something. So just collecting information itself is going to be useful down the road. Whether, I don't know what the company's how it's advertising itself, whether it's useful immediately, I think is a little bit less clear, but certainly it's going to be useful eventually. Okay, thank you very much for coming. It's great.